Can you all see me? Can you all hear me? Yes, people? Can you tell me in the chat box? Yes? Okay, there's a lag. Okay. I guess you can see me now. There's a small lag. Yes? Can you all let me know? Let me know. Hello. Good afternoon. Good afternoon, everybody. Hi, Srimati. Hello, Viveha. Hi, Pooja. Hi, Sushamjot. Very sorry. Oh, we've got Secret Ninja here. Superb. Okay. Okay. Hello, Anthony. Good to see you all. Okay. Right. Okay. So, since Shreyas sir's work is over as physics is done, okay, I guess you had Wasim sir's chemistry yesterday. Yeah? So now we're going to have one-shot series for botany, and then after I do my botany part, Sindoor ma'am will do her zoology part. Okay? Cool? Right. So, now you tell me, how are you people feeling? How are you people feeling? Tomorrow is your exam. What are you going through? Huh? What are you going through now? Tell me how you're feeling. Hi Deepika. Hello Kirtana. Hi Lekha. Hi NNMM. Hi Ecclesia. Hi Abhinaya. Hello Janu. You are afraid of tomorrow's exam. Hi Samuel. Hello Srimati. Cool. Okay, Kirtana is cool. Very good. See, the chats might be a little slow. You might hear me calling out your names a little later. But yeah, nervous. Can't say. Okay, fine. Anxiety. Very scared. A lot going inside. Very... I can't even read out that emoji. Okay. We want Wasim sir. Arey, Wasim sir session is over na yesterday? Okay. I'll ask him to come again. Okay. Thank you, Tamim. Going through 5 stages of depression altogether, Astrophile. Mood swings, scared, anxious. Okay. I know it is very difficult because there's a lot of negative kind of emotions. Okay. Too many negative emotions going inside your head. I understand that. Okay, you're all scared, you're all nervous, but a little bit of that is enough. Okay, a little bit of nervousness is good. It is going to help you to focus. Too much is not good. Okay, too much is not good. So practice some breathing exercises. Okay, control. Make sure that your breathing is in your control. It'll help a lot. I'm telling you. Okay, for tomorrow it will be helpful. Right? It's just a test. Superb. It's just a test. Exactly what I'm talking about. Right? Hold everything together. Just we have one more day to go. Okay, so tomorrow by this time you should all be in the center, or at least almost reaching the center. Okay, it's 1 o'clock, no? So tomorrow you should almost reach the center, reach early. Plan of reaching the center early, and get to know first, understand where it is and everything. Okay? Yes. How to reduce negative marks. You've been practicing all this while, no? So that will help a lot. Okay? You practicing questions will definitely help to reduce the negative marking, because that's how you create, okay, this I don't know, this I know, this I don't know. So you should be able to actually judge the question. Okay, this I know. If you know, you give the answer. If you have no clue of the answer, just don't say, okay, you know what, I'll just give something and come back. No, don't go for that. Okay? Right. Absolutely. So I have put up, or my content team, we have put up a huge PPT with a lot of questions which are prone to come. Okay, for the exam tomorrow, we will go through those questions. You guys can answer in the chat box. Okay, we just slightly out of focus. Okay, it was fine earlier. Let me see. Okay, try reading the question. I'll try fixing it. Genera like Selaginella and Salvinia produce 2 kinds of spores. Such plants are known as. You start the quiz. Let me just fix it. Okay? Should be better now. It should be better. Yeah? B, D, what? Which one? Huh? Okay, so when they are having two different types of spores, they are called as heterosaurus or heterosporus? It is heterosporus, no, because they are talking about spores. Yes, so it should be heterosporus. Okay, easy question. This was just to warm you up. Okay, this was just to warm you up. Easy question. Right? Okay, so the answer is heterosporus. Next question. See, all these questions are going to be easy. Tomorrow's questions are also going to be easy. Okay, don't worry. Dobson units are used to measure the thickness of what? Dobson units. Yes, people. Thickness of CFC, stratosphere, ozone, troposphere. Absolutely. That's how we determine, no, what kind of questions do come in the next year. Yes, very good. Very good, very good, very good. Okay. Right, so Dobson units are used to measure the thickness of ozone. Yes, we know that in the stratosphere, we have a layer of ozone protecting the entire planet from the harmful radiations of the sun. That is the UVA, UVB and UVC radiations. Yeah? Okay, so the answer is C. Next question. Mutations in plant cells can be induced by. See, by this time you should all be knowing the question. Okay, so I'm not going to ask you to even give me any emojis in the chat box. Okay, but you can definitely start liking the video. Yes? Mutations in plant cells can be induced by what particularly? Yes? Not all of them are PYQs. Some of them are sample questions or questions that we feel are going to come for the next exam. Okay? So, kinetin and zeatin, they are plant growth regulators. They are not mutating or mutating agents or mutants. Okay? So, they are hormones. It's either gamma rays or infrared rays. Yes? Pakka? Gamma rays? Okay. Right. Right. Okay. So, I think we will go for gamma rays. Okay? The answer is gamma rays. Right. Next one. The centriole undergoes duplication during what? Yes, the centriole undergoes duplication during? Absolutely. These are the type of questions. Actually, we have done a lot of revision of questions, okay, which are prone to come tomorrow for the exam. So, go ahead. Let me see if I can do it. Yeah. Which phase? When do you think centrioles also undergo duplication? Superb. The answer is S phase. Okay, so during the synthesis phase, it's not only DNA which is getting synthesized, it is also the centrioles getting replicated. Okay, so that it is equally given to the daughter cells. Simple questions, very easy questions. Next one. Conifers are adapted to tolerate extreme environmental conditions because of? Yes, Yamini. Conifers are adapted to tolerate extreme environmental conditions. It can be extreme heat or extreme cold, whatever it is. Yes? Let me see. I'll try to reduce the brightness, okay? Okay, right. B. Thick cuticle. Okay, I agree with you people because vessel superficial stomata is stomata on the outer side. Okay, it is not going to help in reducing water loss, or it is not going to help in help in creating an extra protection or anything. Okay, so yes, The answer is thick cuticle. Okay? Superb, 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 superb. Okay. Right, next question. This amongst the following facilitates the opening of stomatal aperture. Which one of these? Which one of these facilitates the opening of stomatal aperture? Okay, they'll come to fix the brightness. Okay, you can give the answer. I've called them. They'll come to fix the brightness. Don't worry. Right. Just till then, stay with me. Okay. So radial orientation of cellulose microfibrils. So in the stomata we do have guard cells. Okay, and generally in dicots we have kidney-shaped guard cells. So they have a very thick inner wall. Okay, the inner wall is going to be very thick and the outer wall is going to be quite thin, right? At that time, they will have something called as radially arranged microfibrils. Okay, so when they are radially arranged, what happens is, due to turgor pressure, it will pull the pore inside. Okay, the stomatal pore inside will be pulled so that it opens. Yes, so it is important they have these radial kind of lines or strings or whatever you want to call it. Okay, so microfibrils basically, but yes, they help in opening of the stomatal pore. Okay, next question. The answer is C. You guys are talking already. Okay, gemmae are present in which of these organisms? Hello, Eason Muggle. I'll get the brightness fixed, people, don't worry. Yes. Where do you find gemmae? Gemmae are vegetative propagation structures inside gemma cups. Okay, so we have specialized gemma cups, and where do you find these gemmae? Yes, people. In mosses. Why do you find them in mosses? Mosses, there is no place actually to have the gemma cups. Okay, so gemma cups are actually when you have the liverworts. Okay, you have these small cup-like structures. Okay, inside which, inside the cup structure you'll have these small gemma cups. Okay, I mean gemmae structures. So if this cup is going to fall off, onto the soil, it is going to grow into a new plant. Okay, yes, Darun. I know. I'll get it fixed, don't worry. Okay, so it's going to be found in liverworts. Can you give me an example of liverworts? Yes, Anushka, absolutely. You can expect these kind of questions. Okay, these ones or related ones, obviously. Yeah? Okay, I'll use black now. Can you give me other examples of liverworts? Okay, Markantia, yes, absolutely. Markantia is there. Okay. So, liverworts are a group which contains example Markantia and also Rixia. Okay, there's another Rixia also, but yes. Okay, right. So, we'll go for liverworts. Okay, very good. Next one. In spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? Okay, which is a mechanism they would have started? They'll be like, okay, we are fighting too much. Okay, we have to reduce this fighting. We have to all live in peace. Okay, so they would have come up with some compromise. Okay, what is that compromise here? Sphagnum is a moss. Okay, Ashwin, sphagnum is a moss. It's not, what do you say, bryophyte. Yes, Bavadarani, we'll see. Okay, so compromise. Yes? Okay, so their compromise is by resource partitioning. Okay? By resource partitioning, they'll compromise and say, okay, you know what? Okay, enough of fighting. Enough of fighting. You take this much of area. This is yours. This is mine. Okay? Resource partitioning. Superb, superb, superb. Yes? Also do click on that like button. Yeah, come on. You guys are giving such amazing answers. Right. So, resource partitioning is the answer for compromise. Next one. Which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? Okay, here we are going to stop the chiasmata. Okay, there is chiasma formation. We are going to break it and we say, okay, you stay separate. Where is it? We are having a Tamil session, Yamini, later. Okay? At 5 o'clock. Okay, so in which stage of meiosis do we have terminalization? Terminalization is particularly talking about ending. Okay? Ending or stopping of chiasma formation. Yes? It is happening during diakinesis. Okay. Now you guys tell me. Do you feel, okay, after giving these questions, do you feel confident? Do you feel like, okay, this is easy, this I know, okay, this is fine? Do you feel like that? Yes or no? Oh, all correct, so green heart. Why not? Why not? Today is special. Definitely green heart for you. Yes? Do you really feel? See, Srinivasan, you will definitely find these level of questions only. Okay? There's nothing difficult going to come for the exam tomorrow. Okay? This should be increasing your confidence. Okay? It should be because now, let me tell you one thing. It's not because these questions are easy. Okay? I'll tell you, these questions to a normal 11th, 12th student might not be so easy. It's easy because you have been doing all the hard work. Okay? It's easy because you have already studied this. Ma'am, such easy questions. I've already done this. Okay? I've already practiced these questions. I've already studied these concepts. Okay? It's not because the questions are easy. It's because you are prepared. Okay? It's because definitely. Okay? See, if the level is too low, you people are telling me ki these are all previous year's question, and you are only telling me that the level is too low. So what are you saying? That the questions they ask in NEET are low level questions? Yes, that's the best example. No, when you're doing previous year's questions and you feel that this is easy, definitely, okay, definitely that means tomorrow also there are going to be very easy questions. It's just that when you are tensed, you might not find it easy. Yeah? Moving on. So yes, your answer is right. It is during diakinesis or at the end of prophase 1 that the chiasma formation is going to be terminalized because crossing over is done. Okay, recombination has happened. They've all got each types of genes and it's done. Okay? Right. Which of the following algae produce carrageenan? Now this is a repeated question. Okay, this is a repeated question. They ask this question almost every year. Okay, so go ahead, give the answer here. Which of the following produces carrageenan or carrageenan? Okay, yes, superb. Okay, so one thing you always have to remember is that carrageenan, okay, is found in red algae. Okay, it produces carrageenan, and brown algae produces algin. Okay, they produce algae. What are these? These are actually hydrocolloids. Okay, these are hydrocolloids produced by these particular algae, and it is important. Okay, algae is going to be produced by brown algae and carrageenan by red algae. Okay, super. Next question. Hello Nishwant. The factor that leads to founder effects in population. Yes. Used for making agar is not this one. Okay, to make agar we use Gracilaria and Gelidium. Yes, Ranjana, Tirumurthy. That is gelatin. Gelatin is for different purpose. Okay. Gelatin is made by Gracilaria. Okay. And agar is also made by red algae. Okay. Hydrocolloids are different. Hydrocolloids are different. Okay, you give the answer for this now. Who or what leads to founder's effect or founder effect in population? Yes? Come on. If my class is nice, come on now. Give me your likes also. And some green hearts in the chat box. Yes. Genetic drift. Okay? It's a very direct question. It's a very direct question. Genetic drift, when it happens, it leads to the founder effect in population. It happens when we take several population, or specifically we take population into consideration. Okay? Next one. The term used for transfer of pollen grains from anthers of one plant to stigma of a different plant during pollination brings genetically different types of pollen grains. Okay, what do you call it? What do you call it? Okay. Pakka? I'll lock this. Option A? Yes? Okay, so let's revise this as we are going by. Okay? So, xenogamy is basically cross-pollination. Okay? Okay, here pollination is going to happen between different plants. Okay, pollination is going to happen between different plants. Geitonogamy is going to be self-pollination only. Basically, it's going to be self-pollination, but here it's happening between the flowers. Okay, different flowers maybe. Okay, different flowers, but same plant. Okay, different flowers on the same plant. Okay, that is geitonogamy. And chasmogamy are basically open flowers or open normal flowers. And cleistogamy are closed flowers. Okay. So please know all of these. See, whenever it comes to different names, they like to experiment. Okay, the same way for pollination. Okay, that's also very likely asked question. What is ornithophily? What is entomophily? What is malacophily? Okay, those kind of questions also can come. Right? Those kind of questions can also come. So, be prepared for pollination also. Right? Okay, so here the answer is xenogamy, that is cross-pollination. When the pollen grains from the anther is taken to the stigma of a different flower on a different plant completely, and it induces variation. Super. Next one. The amount of nutrients such as carbon, nitrogen, phosphorus and calcium present in the soil at any given time. Yes, at any given time you take the inorganic substances in the soil. What is it called as? What is it called as? Okay, thank you so much for the green hearts. But I also need answers. Yes, come on people. That's very sweet of you. Yes, you give me the answer for this, I will. Yes. C, standing state. Okay. So, I have always learned standing state and standing crop is a little, what do you say, confusing. Okay, always remember state as in state in chemistry. Okay, states of matter in chemistry. So, we have actual proper inorganic substances. Okay, inorganic chemical substances in the soil at the right time is called as standing state. Now, standing crop is more like bio-related, okay, or agriculture-related. So, the amount of organic matter, okay, the amount of organic matter in a given time at a given place, right? So, standing state you relate to chemistry or inorganic chemistry, but when it comes to crop, go for bio, which is our organic matter. You can also call it as biomass. Okay, you can also call it as biomass. Okay, so living matter we go for standing crop, and inorganic substances we go for standing state. Perfect. Okay, so standing state is the answer. Next one. Which of the following algae contains mannitol as reserve material? Please, 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 whatever you want to revise in the last moment, please revise the table. Okay, table inside your NCERT book for algae. It's very important. Yes? I'll get it fixed, people. I'll get it fixed. Don't worry. Just hold on a little more. Okay. Which of the following algae contains mannitol as reserve food material? Very easy question. Okay. So we have mannitol and laminarin always comes together. Okay. Mannitol and laminarin. Okay. Both of them are stored food of brown algae. Okay, they are all stored food of brown algae, and here the example for brown algae is Ectocarpus. Okay, the example for brown algae is Ectocarpus. Okay, perfect, 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 perfect. Okay, the answer is A. Right? Next one. Which of the following is not a product of light reaction or photosynthesis? Not a product of light reaction or photosynthesis. What do you get as a result of light reaction? Or non-cyclic photophosphorylation. We generally take non-cyclic as light reaction. Okay, so go ahead. You don't have to be scared. I'm telling you. Okay, Pakka. See, tomorrow we will be doing the paper discussion also. Okay, we will start the paper discussion, and then I will tell you how many questions that I thought have come for the exam. Okay? So yes, wait for tomorrow. I am also waiting for tomorrow. If there are questions coming out from this, no, then I am going to come and pull your ears. Okay? Which of the following is not a product of light reaction? ATP, NADH, NADPH, oxygen. So, here we have NADH, NADPH. Usually, NADH is found in respiration. Okay, it is during respiration that the body produces or the cells produce NADH. In photosynthesis, they prefer NADPH. Okay, they prefer NADPH with a phosphorus in them. Right? Okay, so this is a very direct question. So, in light reaction, we do get ATP, NADPH and oxygen. Okay? Now, I have a question for you people. Okay, don't look at the screen. I have a question for you people. This is what you get. These three you get in non-cyclic photophosphorylation. What is the product you get in cyclic photophosphorylation? Yes? What do you get? What do you get in cyclic photophosphorylation? Okay, Darun. What do you get? What do you get as a result of cyclic photophosphorylation? NADPH? Yes, NADPH, NADPH plus, yes. Okay. ATP only, pakka. Superb, superb, yes. So we get only ATP. Okay, now the brightness is perfect, right? We get only ATP. Right. Superb. You'll all try and get 360 on 360 in bio. Okay? This question. Which of the following stages of meiosis involves division of centromere? Yes, which involves division of centromere? Okay? In which phase do you have division or separation of centromere? That means, yes. So basically, when it involves this division of centromere, we are going to separate, okay, we are going to separate the sister chromatids. Yes? We are going to separate the sister chromatids. Right? So where does it happen? Separation of sister chromatids happen in anaphase 2. Yes? Anaphase 2. Right. Perfect. Because that is when in first round of meiosis, okay, or in meiosis 1 we have separation of homologous chromosomes. We are not separating the centromere. It's just that we are separating the homologous chromosomes. Okay, they're separate chromosomes. But when it comes to meiosis 2, they're like, okay, now we don't have anything more to separate. We have to separate the sister chromatids. Okay, so they have to separate the sister chromatids or the centromeres in anaphase 2. Next one. The ovary is half inferior in. Okay, the ovary is half inferior in which of these plants? Which of these plants? Board was not clear. Yes, Lucifer, absolutely. Now it's clear, no? It's clear now. Sunflower? No. Okay, let's relate and learn. Okay, so half inferior ovary would be a perigynous flower. Okay, it would be a perigynous flower. Okay, so perigynous flower example would be something starting with P and something with R. Okay. Plum, rose and peach are the examples of perigynous flowers. Okay, because it has P and R, it's easy to remember that way. It's easier to remember that way. Okay, so half inferior is perigynous flowers and the answer is going to be plum. Okay, yes, perigynous flowers, half inferior ovary, perfect. Okay, tips to solve MCQ. Okay. See, basically, these kind of questions, you have to go to the direct answer, okay? Now, this is what I would call a proper example-based question, okay? So, when you're getting an example-based questions, you don't have to think much. If you know the example, you know the example. If you don't know, you sitting and looking at the paper like this, you're not going to get the answer. No way. Okay? But when you have conceptual questions, when you have like real conceptual questions, that will, help you. Okay, this I can maybe think a little bit, read the question a little more, get the details, and then, answer the question. Okay? Now, when I find questions like that, I will tell you. So, this is the direct question. If you know that perigynous example is plum, you know it. Otherwise, it's wrong. There is no other easier way. Okay? Now, this question. Which of the following regarding post-fertilization development is incorrect? Okay, so here maybe you can think, right? You can think and you can find an answer here. Let's see. Ovary develops into fruit. Zygote develops into embryo. Central cell develops into endosperm. Ovules develop into embryo sac. Right? So now here you can use all the concepts that you have learned. Whatever concepts you have learned about post-fertilization events, okay, that is, we all know that that the new cell is, okay, new cell has one particular cell called as a megaspore mother cell, okay, that is going to form the embryo sac. Yes, that's going to form the embryo sac. Okay, we know this. Now, the same way, if we talk about post-fertilization events only, we can say that the zygote will form the embryo. Okay, and we have the primary endosperm nucleus, okay, or inside the primary endosperm cell, okay, they will form the endosperm. Okay, now this endosperm and embryo together, will form the seed. Okay, it will form the seed. Right? Now, ovary into fruit, ovule into seed. That is something we all know. We have been learning for a very long time that the ovary, okay, or the ovary wall develops into the fruit or the fruit wall. Okay, or pericarp. Yes, everything is related. Everything you can link from one chapter to the other. Then, ovule at the same time will develop into forming the seed. The ovule, which contains embryo and endosperm, will form the seed. Okay, so when you have a question like this, you can actually relate the entire chapter and you can say, okay, ovary develops into fruit is right, zygote develops into embryo is right, central cell develops into endosperm, perfect, because here this is our central cell. Okay, this is your central cell which develops into the endosperm. Ovules develop into embryo sac? No, ovules are the result. Okay, so automatically this is a conceptual question which you can spend time in thinking and you will eventually get the right answer. Yes, it's not like any other example based question. Right? Next one. Okay, somebody asked me haploid diploid condition in ovule. So basically the zygote, okay, and all the mother part cells, no, they are all going to be zygote, embryo, then new cell is, okay, they are all going to be diploid. Okay, they are all going to be diploid. Now what are the haploid cells? The haploid cell is just the egg, okay, egg cell and all the cells inside your embryo sac. Okay, basically each of the cells or each of the nuclei of your embryo sac is going to be haploid. Okay, then we have triploid also. Yes, we have triploid also. Primary endosperm nucleus, primary endosperm cell, okay, or basically your endosperm in angiosperms only. Okay, in angiosperms the endosperm is triploid. Right? Now, can you tell me what is a ploidy of endosperm in gymnosperms? Yes? Do you people know? What is the ploidy of endosperm in gymnosperms? Is it haploid, diploid or triploid? Yes? In gymnosperms, it is haploid. Yes, very good. And in angiosperms, it is triploid. Okay, in angiosperms, it is triploid. Okay, this is also a possible question, or something related to this can also be a possible question. Okay, how many questions I don't know. The questions based on diagram will usually be labeling. Okay, they'll give you the label A, B, C, D and they'll ask you which is A, B, C, D from the options. Okay, yes, half inferior. There is nothing called as half superior, Antony. There is only half inferior. There is no half superior. Okay. Next question. Name the plant growth regulator upon which spraying on the... Okay, so we are going to spray a plant growth regulator onto sugarcane crop. It increases the length of the stem, thus increasing the yield of sugarcane crop. Okay. So here you can recollect everything we learnt about internode elongation. Okay. So internode elongation we learned in grapes. Okay. Sugarcane. Then what else? Bolting. Okay. Faster malting. Okay. And yes, breaking seed dormancy. Yes, this is how revision should be. When you get one question, you relate everything maximum possible to that question. Okay. Perfect. 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 Yes, you guys are right. It is gibberellin. Okay. Gibberellin increases internode length. That's the main function. Okay. This one and this one is the main function of gibberellin. Super. See? You guys are getting better, people. You guys are getting better. Don't worry. Okay, be confident. You will do this. Right? Hi, Abhi. Okay, gibberellin. Next question. In some plants, the female gamete develops into embryo without fertilization. This phenomenon is called as... Easy question. Easy question. Yeah? Right. Come on. I know it's a little lag, okay? Me reading your answers, it's a little slow. That's why I'm going too slow. It feels like I'm going too slow. You're giving the answers and after 10 seconds only I'm replying for the answer. Okay? It's a little slow. Anyways, you guys are all right. The answer is parthenogenesis. Okay? Parthenocarpy is forming the fruit. Yes, no fertilization or forming the fruit before fertilization. But in those conditions, there is no, there is no embryo, there is no seed. Okay? If they are mentioning embryo, then it should be parthenogenesis or apomixis. Actually, you can give, if the option of apomixis is there, you go for that. Okay? Please go for that. Right? So, when they have mentioned female gamete develops into embryo, that means there is a seed. Okay, the seed is formed without fertilization. That you will give as apomixis. Okay, apomixis or parthenogenesis. Yes, they are quite relative terms. Okay, out of these options, let's go for parthenogenesis only. Okay, parthenocarpy is fruit. Okay, fruit formed without seed. Okay, that is some kind of miscommunication. So usually what happens, okay? Oh my god, rebellious children. Seriously. You want to start preparing for NEET 2022 today within one day. Okay, you know what? I'm not going to answer that question, rebellious children. The other students, please let this person know if they'll be able to score 650 above if they start today. Okay? I'm not going to answer that question. Anyways, so I was talking about... How does parthenocarpy happen? It is actually... Okay, so it actually happens when the ovule is undergoing fertilization, it usually gives a signal saying, okay, hello, listen, ovary, you can become a fruit. Now, if the ovule sends a wrong signal, or the ovary is so preoccupied that it is like, okay, I think fertilization happened, I think I got a signal from there, okay, let's start forming the fruit. Yes, so that time they're going to form the fruit and then later realize that, oh my god, there's no seed. Okay, because the ovule was not fertilized. That's when you get parthenocarpic fruits, seedless fruits. Okay? Next one. Okay, the product or products of reaction catalyzed by nitrogenase enzyme in the root nodules of leguminous plants. C and D ka meaning. Syngamy, autogamy. Syngamy is what? One second, I'm very sorry. I just saw the message. Syngamy is when the male gamete, okay, male gamete fuses with the egg cell, okay, you call it as syngamy. Autogamy is self-fertilization. Okay, somebody asked this, so anyways, you give me the answer for this. What are the products you get after reaction of nitrogenase enzyme? Okay? Absolutely, Ashok. Okay. Ammonia and hydrogen. Okay, so nitrogenase is a very special enzyme that are found only in prokaryotes. Okay, the plants don't have nitrogenase. The plants feel that it's too below its pay grade. Okay, so the plants will not have nitrogenase. It will hire prokaryotes which have nitrogenase enzyme, and they'll say, you do the work. I'm not going to do it. Okay, you do the work for me. So the prokaryotes have nitrogenase enzyme and they will convert atmospheric nitrogen into ammonia and hydrogen. Okay, so usually these are the products that is produced. It's a direct question. It's a direct question. You can't make ammonia oxygen. You can't make nitrate alone. Okay, then the only confusion is, do they make ammonia alone or ammonia and hydrogen? Here you have ammonia and hydrogen. Okay, next one. Yes, Akash, you can. If you want to, you can. Which of the following is the most important cause for animals and plants being driven to extinction? What causes extinction? Or loss of biodiversity? Yes? Very good. Okay. See, alien species invasion is not going to be that effective. It is working. Yes, I'm not saying it's not working. But more than that, habitat loss and fragmentation. Okay. That is pretty much destroying the homes of organisms. You destroy the homes of organisms, you're destroying the organism by itself. Okay. So that is considered the most cruel way, or the most, established way of reducing the biodiversity or driving organisms into extinction. Yes? Right. Next question. That is also a direct question. In light reaction, plastoquinone facilitates the transfer of electrons from. Yes? Now you can't say all these questions are easy. The level has increased. Yes, you give me the answer for this one. Come on, people, and also drop some likes. Yes, look at the likes. The people watching are double. Yes, awesome, awesome. So, plastoquinone is going to be formed between PS2 and cytochrome B6F. Okay? So, just remember the flow. Just remember the arrangement. Okay? I would prefer you people remember the arrangement. So, it will start from PS2. Okay? You can learn that there is somebody in between called as Yeah, definitely Plastoquinone is there. Pheophytin you can remember if you can. Okay, we have pheophytin. Then we have Plastoquinone. Okay, and from Plastoquinone to cytochrome B6F. From cytochrome B6F it goes on to plastocyanin. From plastocyanin to PS1. Okay, from PS1 it moves ahead to ferredoxin. Ferredoxin to FNR. Actually, the electron does not move to FNR. Okay, it helps in the production of NADPH. So it works with NADP plus electron plus H plus, and you get NADPH. Okay, remember the flow of electrons. So remember what are the proteins present. This will help you a lot. Okay, more than the processes, this will definitely help you. Okay, still easy level questions. Okay, offsets are produced by what division? Offsets are produced by what division? Such easy questions, but I'm still getting wrong answers. How is that? Huh? Okay. So, offsets are a type of vegetative propagation. Okay. So, they are a type of vegetative propagation. Patho, Carpe, no. Meiotic divisions will happen only in sexual reproduction. Okay, only in sexual reproduction we can have meiotic divisions. Parthenogenesis also no. These are all deviations. Okay, the best answer would be mitotic divisions because it's directly forming from the parent. Okay, asexual mode or vegetative propagation. So mitotic divisions. Next one. Which of the following statements is correct with reference to enzymes? This is the easiest question. Okay, I agree. Okay, this is very easy question. Let's see. Yes, Ragul. Answer for this. Answer for this. Come on, people. Right. The answer is holoenzyme is equal to apoenzyme plus coenzyme. Okay, this you guys have done it. Very good. So this is direct. Okay, there's nothing for me to explain. Apoenzyme. Now holoenzyme is a whole thing. Okay, we all know. For example, coenzyme or co-activator, or for example, let's take pyruvate dehydrogenase. Okay. Yes, this is also not visible. Okay, when we take pyruvate dehydrogenase, okay, we need a coenzyme or we need a cofactor Mg2+. Okay, that is a cofactor. Like that, okay, some enzymes, some of the apoenzymes also require an additional coenzymes. Like for example, coenzyme A. Okay, let's talk about coenzyme A. Okay, so coenzyme A is there. Yes, coenzyme A is there to make acetyl-CoA. Okay, so coenzyme A is a coenzyme along with whichever, whatever the enzyme is. For example, citric acid. Okay, to make citric acid, we might require citrate synthase. Or for making acetyl coenzyme A, we have dehydrogenase, pyruvate dehydrogenase along with coenzyme A. Yes? So definitely coenzyme and apoenzyme together. Okay? Perfect. This is a very direct question. Next one. The first stable product of carbon dioxide fixation in sorghum is. Very easy question. Go ahead. Yes, people. How many questions can we expect? From where? We have the weightage, no? The weightage is given online. So, yeah. Perfect. First stable product of carbon dioxide fixation in sorghum. Sorghum is a C4 plant. Okay? Sorghum is a C4 plant. Yes? So, in C4 plant, the first product is oxaloacetic acid. Again, direct question. Okay? Next one. The region of biosphere reserve which is legally protected and where no human activity is allowed is called as So in a biosphere reserve we generally have at least 3 levels. We generally have at least 3 levels. Okay? So where there is no human activity? Perfect. Yes. Yes. So this is your transition zone. Okay, this is your buffer zone. And this is your core. Okay, core zone. In core zone we do not allow any human activities. Yes, in the core zone we do not allow any human activities. In buffer zone and in transition zone we can allow. Okay? Buffer zone mein we allow only researchers and educators. Okay? Transition zone mein we can have people living. No problem. Next one. Okay? Identify the basic amino acid from the following. Yes? Which is the basic amino acid from the following? Come on, people. A. Superb. Yes. Lysine is a very, why is it basic? Because the side chains have a positive charge. Okay, the side chains of lysine and arginine and all, okay, and histidine, okay. So there are 3 amino acids which are basic. Okay, there are 3 amino acids which are basic. Okay, lysine, arginine and histidine. Okay, they're all basic amino acids because they have a side chain. Yes, from this section. I didn't understand which section, Akshita. Okay, so they all have side chains containing a positive charge and that is why they are basic in nature generally. Okay, next one. Which of the following statements is incorrect? Which is incorrect? Which is incorrect here? Read. ATP synthesized through complex 5 oxidation reduction reactions produce proton gradient in respiration. During aerobic respiration, role of oxygen is limited to the terminal stage. In ETC, 1 molecule of NADH gives rise to 2 ATP molecules, 1 FADH2 gives rise to 3 ATP molecules. Superb. From this session, oh, it depends. See, we have taken a lot of PYQs. Okay. I can't tell you how many questions will come here. I'm not setting the paper. I'm just giving you maximum type of questions, okay, which we expect. We think that, okay, some of these might come. I can't give you a number properly because I'm definitely, if I was setting the paper, I would have told you, like, properly, yes, this will come, this will come, this will come. I would have told you. Not possible, no? Yes. Superb. Okay, so generally, NADH will give rise to 3 ATP molecules and FADH2 will give rise to 2 ATP molecules. Okay, that is why here they have given it ulta, and that's why this is incorrect statements. Yes. Next one. We'll finish it with another 5 minutes, okay? We'll finish the session within another 5 minutes. Which of the following organisms are known as chief producers in the ocean? Direct question, direct statement from the NCERT. Okay? Direct statement from the NCERT. Physics session is over. Next you will have zoology session with Sindoor ma'am. There are many wrong answers coming up. Okay. The right answer is diatoms. Okay. Diatoms are known as the chief producers of the oceans. They grow immensely. Okay. They are generally, unicellular organisms. But they grow so much in the oceans that they are considered as the chief producers. And they photosynthetic also. Okay. Easy. Next one. Strobili or cones are found in. Where do you find strobili or cones? Yes? Perfect. Okay, yes. Equisetum and Selaginella. Okay, so in Equisetum and Selaginella, you have specialized structures called as strobili or cones, where we have these sporangium and the sporophyll arranged compactly. Okay, perfect. Next one. Since you want me to end it fast, we'll go fast. Life cycle of ectocarpus and fucus respectively are. This is a question from alternation of generation. Okay, yes, you are doing right. Perfect. Life cycle of ectocarpus and fucus are. You get this answer right, I'm giving you some green hearts. Yes? Perfect. Okay? This we have learned as exceptions. Okay? We have learned this as exceptions. So, we have ectocarpus, we have Polysiphonia, we have kelps, which come under haploid diplontic life cycle, and we have fucus, although these are all algae, they come under haploid diplontic and diplontic life cycle. Okay? Perfect. Green hearts for you people. Next one. Okay, so here. And other than that, gymnosperms and angiosperms are also diplontic. Anyways. Which of these statements is incorrect? Glycolysis operates as long as it's supplied with NAD that can pick up hydrogen atoms. Glycolysis occurs in cytosol. Enzymes of TCA cycle are present in mitochondrial matrix. Oxidative phosphorylation takes place in the outer membrane of the mitochondria. RS Madan FF Tamil class at 5 o'clock. Perfect. Yes. So, we can say the answer is D. Okay, why? Oxidative phosphorylation takes place in mitochondrial membrane, okay, but not in the outer mitochondrial membrane, okay. Specifically, it is in the inner mitochondrial membrane. Okay, it takes place in the inner mitochondrial membrane. Very simple, all of this are perfect. Okay, glycolysis operates as long as NAD is supplied because they are making NADH. Okay, it occurs in cytosol, perfect. Mitochondrial matrix, yes. Okay. Next one. Which among the following are the smallest living cells known without a definitive cell wall, pathogenic to plants and animals, and can survive without oxygen? Yes? Absolutely, Kaveri. I agree. No, Sachin, different questions. Yes? Perfect. Mycoplasma. They are also called as the smallest living cells. Okay? Mycoplasma and PPLO. Yes, if you see Mycoplasma and PP yellow, okay, Pleuropneumonia-like organisms, they are also the smallest living organisms. Okay, perfect. Next one. Which of the following events does not occur in rough endoplasmic reticulum? Which is not there in rough endoplasmic reticulum? Easy peasy lemon squeezy. Thank you people, thank you so much for the hearts. A little likes also please. Yes, cleavage of signal peptide, protein glycosylation, protein folding, phospholipid synthesis. Something that does not happen in endoplasmic reticulum. Yes, the answer is D. Phospholipid synthesis happens in the smooth endoplasmic reticulum. Okay, it takes place in the smooth endoplasmic reticulum. Lipid of any kind is formed in the smooth endoplasmic reticulum because all the other protein related stuff is happening in the rough endoplasmic reticulum. Okay, perfect. That means, yes, we are done. Again, this is for people who are still, going to listen to concept videos and try learning the concept. We do have the CC Lite for you. But again, I don't think you need this. Okay, if you have any other queries regarding any of your courses, you can WhatsApp to this number. It's there. Okay, and yeah, please do like the video. I will call in Sindoor ma'am now. Okay, and I can't say don't worry to you people. I know you'll all be worrying. You'll all be very scared. Okay, don't think the questions that I showed you was very easy. Okay, that is the level of questions you will have for NEET. The PYQs have been taken to prove that only. Okay, the PYQs are PYQs. That is, they have been asking such easy questions in NEET, and that's exactly what I was showing you. Okay, so please don't think that these are easy questions. Okay, tomorrow we're going to get more difficult questions. No, you won't. Okay, right. Thank you people. All the best for your session. For your exam tomorrow. Okay, you will always have our blessings and our prayers. Always. And yes, we are also a little nervous. Okay, regarding your results and everything. So just go and do your best. Okay, give your best. Right? Thank you everybody. And yes, all the best. All the best to all of you. Okay, in Tamil class we won't have the same questions. We have different type of questions there. Okay, yes Lucifer. Absolutely Lakshmi, thank you. Take care all of you. All the best. Bye Satvik, Vishnu, Susie. Bye Hot Stuff. Bye Basil, Shivakumar. Bye Gautam, Abhishek. Take care all of you. Bye bye. All the best. Bye people.
Hi everyone, am I audible? Am I visible? Hi everyone, very good afternoon. How was the session since morning? How was your session since morning? Are you guys ready for Sureshot zoology questions? Is everyone ready for Sureshot zoology questions? The last day Sureshot most expected questions for NEET 2022. Is everyone ready? Very good afternoon everyone. जल्दी से आ जाओ, जल्दी से आ जाओ. Very good afternoon everyone. Everyone ready? Okay, perfect. So yes kids, let's begin. Alright. Chalo, you will be answering everything on the chat box. For those of you who are new to the channel, I'll quickly introduce you to myself. I'm Dr. Sindhu, NEET Zoology expert at Vedantu. I have been teaching for NEET Zoology from past 10 straight years. Very good afternoon. Any of these questions in either the same way or in just a little twisted way you can expect in your NEET exam tomorrow. Yes everyone, we will be solving everything today together, so pay attention. This is going to be in English, do not worry. Hi Abhi King. Yes, this will be in English. Amrita, don't worry. Yes. Hi everyone. Yes, I will be taking in English. Do not worry, Kabish. This is absolutely, this channel is made only English. Okay. Chalo guys, tell me the answer. Which one of the following belongs to the family Muscidae? Which one of the following belongs to the family Muscidae? Vishnu, Tamil, no, but English, yes. Okay. Vishnu, Tamil, no, but English, yes. Okay. Yes, everyone. Very good. Is it? Perfect. The correct answer here is housefly. Correct answer here is going to be housefly. Excellent, everyone. Housefly. Okay, that was an easy one. That was a very easy one. Let's continue. But do you know? It was easy, but do you know it was asked previously? It is a PYQ. Chalo, next question. Receptors for sperm binding in animals are present on. Receptors for sperm binding in animals are present on. Where are the receptors present for sperm binding in animals? Receptors are present on. Where are they present on in animals? Come on everybody. Corona radiata, vitelline membrane, perivitelline space or zona pellucida? Yes. Where are the receptors present? Everyone. Receptors are going to be present on the zona pellucida. Very good. On zona pellucida. Quickly kids, let's go through what these layers are. If anyone is having a doubt in that, let us quickly go through which are those layers A, B and C. So, children, this is a structure of ovum in your textbook. Quickly, let's go through this. Okay, everyone, I'm not wasting much time. Here we go. These are the corona radiata, outermost corona radiata cells. The green colored part is your zona pellucida. Green colored part is zona pellucida. Pink colored region is your perivitelline space, and the inner circle you're seeing over here, this circle, the inner circle, this is your vitelline membrane. This is your vitelline membrane. Okay everyone, that is your vitelline membrane. Clear for everyone? Alright, that is what your vitelline membrane is. Alright, chalo kids, next question. Which one of the following is correct? Quickly, very simple question. Which one of the following is correct? Which one is correct over here, children? Which one of the following options here, which one is correct? Very good, Gokul. Perfect, Jayashri. Jayashri, look at the options. Which one here is correct, children? Lymph, blood, plasma, serum. Which one is given the correct constitution? Which one is given the correct constitution? Very good. Blood is equal to plasma plus RBC plus WBC plus platelets. Plasma is how much percent? Plasma is how much percent? 55%. Okay, and all of these come together is how much percent? 45%. All of these coming together is 45%. Okay, everybody. What is serum? Tell me what is serum. Serum is going to be plasma minus clotting factors. Plasma minus clotting factors is what we call as serum. Perfect. Okay. And what is plasma? Plasma is going to be blood minus not lymphocytes but all the cells. Okay. Blood minus the cells, what's remaining behind is plasma. Now from this plasma, when you remove the clotting factors, what you have is serum. Okay. Perfect. Chalo. Next question. Which of the following statement wrongly represents the nature of smooth muscle? They are asking you, read the question very carefully. Are you able to read the question? Is the option visible to everybody? Is the option visible to all of you? Which is perfect, everybody. Following statement is wrongly representing. Please remember, you read the question carefully. Wrongly represents the nature of smooth muscle. Wrongly represents the nature of smooth muscle. Which is this? Are you able to see this? No? Okay. Now are you able to see it, everybody? Sorry. Are you guys able to see it now? All the best, Mithun. Now is it okay? Communication. What is the correct answer? Very good, Kumar Pallav. Yes, that communication among the cells is performed by intercalated disc, not in smooth muscle. Tell me, intercalated disc means which muscle? Yes, kids, tell me. Intercalated disc means which muscle? This is going to be cardiac muscle. Cardiac muscle. This is going to be cardiac muscle. Okay. So that is the wrong statement. Chalo. Next question. In a cross between male and female, both heterozygous for sickle cell anemia gene, what percentage of the progeny will be diseased? Please read genetic questions very calmly. Okay, kids? Tomorrow, the one thing which you need to maintain is your calm. That is the only way you will get through your whole paper without making silly mistakes. Yes? What is the correct answer here? Answer will be C. Very good. First of all, sickle cell anemia is autosomal recessive disease. Correct? It is autosomal recessive. Okay. Also we see male and female are heterozygous, means male and female are carriers. So you have one HbA, HbS. This is the male, and female HbA, HbS. Okay, this is the female. So their children are going to be HbA, HbA. Correct? That is one possibility. HbA, then HbS, HbA, HbS, and HbS, HbS. Correct? So now out of all of these, which one is the diseased one? This is diseased. So 25%. 25%. Okay, everybody? Perfect. Very good. Correct answer is 25%. Next question. Which one of the following organisms bear hollow and pneumatic long bones? Which organism bears hollow and pneumatic long bones? Pneumatic long bones. Which one? Where will you classify pneumatic long bones as? You will classify it as pneumatic long bones. Are you guys with me? Or is chat a little slow? Chalo. Pneumatic long bones is going to be classified under Aves. And the only example here which is of Aves is going to be neophron. Neophron is scientific name of what? Neophron is the scientific name of what? Who will tell me that? Neophron. Neophoron is the scientific name of what? Yes, it is going to be vulture. Yes, everybody, it is going to be vulture. Neophoron is vulture. Correct? Then, yes, what is macropus? Macropus is kangaroo. Okay, what is hemidactylus? Hemidactylus is lizard. Which lizard? Wall lizard. And what is ornithorhynchus? Platypus. Platypus. So correct answer here is going to be answer option A. Very good. Option A. Okay, kids? Okay. Next, during the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, then which of the following steps of PCR will be affected first? If high temperature is not maintained in the beginning, which is affected first? What is the correct option? High temperature not maintained. What will be affected first? Yes, the correct option here is going to be C. That is denaturation. Because denaturation occurs, the breaking of proteins is going to occur only and only when high temperature is applied. Okay. Then when you talk about PCR, the first step is denaturation. The second step is annealing. What is annealing? Annealing means when you are adding oligonucleotides as primers. You are adding oligonucleotide as primer. That is annealing. Last step is going to be extension, where it is amplified billion of times. Last step will be extension, where it is amplified billions of times. Okay, class? Alright. So, yes, the correct option here is C. That is denaturation. Alright. Next question. Which one of the following is an example of hormone releasing IUD? Hormone releasing IUD. Yes, I think my chat is slow, kids. I think so. Yes. Hormone releasing IUD. What is the correct option? Yes. So, over here, 1 copper T, copper 7, multi-load. All these three are copper releasing. All the three are copper releasing. Only and only LNG20 is the one which is hormone releasing. So, the answer here is B. Hormone releasing IUD is LNG20. Yes? Children, 3 types of IUD. Who will tell me in the chat box? 3 types of IUD. Who is going to tell me in the chat box? 3 types of IUD. Who is going to tell me in the chat box? Three types of IUD are 1. Non-medicated. Non-medicated. Second is copper releasing. Copper releasing. And third is hormone releasing. Hormone releasing. Quickly, let's go through their options. Lipase loop. Lipase loop. Then copper T, copper 7, multi-load. And in hormone, LNG20 and progestasert. Kids, this is very, very important. Okay. Next, erythropoietin hormone, which stimulates RBC formation, is produced by erythropoietin hormone, which stimulates RBC formation, is produced by erythropoietin hormone is produced by what? Produced by what? Yes, The correct option here is going to be that it is produced by the juxtaglomerular cells of the kidney. Remember this hormone erythropoietin is a hormone which comes from the kidney. And which part of the kidney? Juxtaglomerular cells. It goes to the red bone marrow and it stimulates that red bone marrow to form the RBCs. Clear? Okay, what do alpha cells of pancreas do? Alpha cells of pancreas are secreting a hormone. Which hormone? Glucagon. Okay, glucagon. Next. So yes, the correct option here is D. Chronic autoimmune disorder affecting neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscle is called as. It is affecting the neuromuscular junction. Yes, everyone, what is the correct option here? It is affecting the neuromuscular junction. Neuromuscular junction. It is going to be C. That is myasthenia gravis. Myasthenia gravis. Now, children, let's just quickly go through this. Okay, when we talk about muscular dystrophy. Muscular dystrophy is what type of disorder? It is a genetic. It is a genetic disorder. It is a disorder, rather it is a mutation taking place on which gene? The largest gene. Which is the largest gene? Dystrophin. Which is the largest gene? Dystrophin gene. It is a mutation which takes place on dystrophin gene, the largest gene. Also, dystrophin located at which one? 2.4 million bases according to your NCERT textbook. Yes, everybody. So, the correct option here is myasthenia gravis. Also, gout is what? Accumulation of uric acid crystals. Uric acid crystals in the small joints. In small joints, accumulation of uric acid crystals is what we are going to be calling as gout. Okay, this is what we call as gout. Alright kids, let's continue. Perfect. So the correct option here is C, myasthenia gravis. Next, DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appear as what? How do DNA strands appear when they are with ethidium bromide viewed under UV radiation? How do they appear? Which color is ethidium bromide going to be giving off? Perfect. Yes, the correct option here is going to be, they will be of bright orange colored bands. Bright orange colored bands, right? This is seen in which technique? This is seen in which technique? We learned this when we learned about PCR. We learned it when we learned all about PCR. Yes, so bright orange colored bands. Alright, now next. Kids, tell me how you finding the questions. जल्दी से मुझे बता दो. Tell me on the chat box, how are you finding the questions? Can you let me know? Let me know how are you finding the questions on the chat box. Let me know, kids. Very good, Tamim. Let me know how are you finding the questions. Are they easy? How are they going? You let me know and I will tell you why I asked you this question. I will tell you in a little while why am I asking you this question. Okay, let's see the next question. For effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? Which is the diagnostic technique used for early detection? Yes, everybody. Diagnostic disease or diagnostic technique used for early detection here is going to be C, that is ELISA. Children, what does ELISA stand for? Enzyme linked. Enzyme linked. Immunosorbent assay. enzyme linked immunosorbent assay ELISA is going to be useful for early detection of any disease. Clear? And what are we using ELISA for? Detection of AIDS. Okay, HIV. Perfect. What is the virus? It is a retrovirus. Okay, retrovirus. And which is the enzyme there? Which is the enzyme? Reverse transcriptase. Okay, and what is the genome? What is the genome? Okay. Next, for effective treatment of the disease. Okay, we're done with this. Sorry, kids. Which of these is not important component of initiation of parturition in humans? Not an important component of initiation of parturition. Not required for starting of parturition. Yes, everybody, quickly. Yes, some people are telling me easy, ma'am. Very good. Okay, no Hindi. Easy peasy lemon squeezy. Very good, Sahiti. I will tell you in a little while why I asked you this question. Chalo, first you answer me this question. Which of these is not an important component of initiation of parturition? And the correct answer here is going to be D. That is release of prolactin. Not required. Not required. Okay. Now look at this chain which I am going to teach you. Quickly I'll teach you the chain. Okay. Look. First of all, estrogen and progesterone ratio is going to increase. Okay. Then oxytocin is going to be released because of the fetus and the placenta. Fully developed fetus, placenta is going to release oxytocin from hypothalamus. Then the prostaglandins is also going to be synthesized because the fetal head is putting pressure on the opening, on the cervical canal opening. Fetal head is putting pressure. So prostaglandins is going to make the cervical canal open, and that is going to help in delivery. But prolactin does not play any role in parturition. Okay, chalo. Next question, everybody. Cuboidal epithelium with brush border of microvilli is found in cuboidal epithelium with brush border of microvilli is found in cuboidal epithelium with brush border. Very simple question, kids. So simple. So simple. Yes, this is going to be in PCT because in PCT, proximal convoluted tubule of the nephron is where maximum absorption is going to take place. That is what we are calling as tubular reabsorption. Yes, Savita, I will answer you. I will answer you. Kids, again I'm coming to why did I ask you this question? Because these are questions which are picked up. They are asked already previously. So do not worry. Tomorrow's paper is going to be just like this. Okay, you are finding it easy because you have prepared well. Am I clear about that? Okay, so please, this is not the time. This is, in fact, I will say that this is the time where you want to believe in yourself, you want to trust yourself, and just keep going. Okay, chalo, next question. In gel electrophoresis, separated DNA fragments can be visualized with the help of, how are you going to visualize the separated DNA fragments? How are you going to visualize the separated DNA fragments? Yes? And perfect. The correct option, yes, Akhil, don't worry. This just means that your preparations are going well. Okay, kids? Please do not think that, oh, ye to bahut easy questions hai. No, this is exactly what NEET expects out of you. So you're doing well. Just keep consistently doing it. Okay, this is your last day. Chalo, ethidium bromide in UV radiation, not infrared and not acetocarmine. Okay, ethidium bromide in UV radiation, everybody. Chalo, next question will come to. Next question, which one of the following statements about histone is wrong? Which one of the following statements about histones is wrong? Wrong statement they want. Okay, kids? Wrong statement. What is wrong over here? The correct... Hi Gokul. No, how can I forget? Are my kids here? Are my Sankalp kids also here? मुझे बता दो. Tell me if you guys are here. Are my Sankalp kids here? Tell me if you guys are here. I will be talking to you all very soon. I will be talking to all of you very soon. Are my Sankalp kids here? Okay. Yes. Which of the following statement about histone is wrong? The correct answer here is pH of histone is slightly acidic. No. No. Histones are coming together to form the octamer. So that has to be positively charged. That has to be basic because it is going to be having the DNA wound around it. And histone forming 8 molecules is going to be your octamer. Okay, and yes, please remember, histone is rich in lysine and arginine. These are the 2 amino acids which are making up histone proteins. And yes, histone carries positive charge. That is why you can put the negative charged. That is why negative charged DNA is going to wind around that histone. And what is that called as? Negatively charged DNA winding around the positively charged histone. What is it called as? Nucleosome. We call that as nucleosome. And when you view it, what appearance is it giving? Beads on a string. Beads on a string appearance. Clear everybody? Very good. Very good everyone. Chalo, next question. Which of the following contraceptive methods do involve a role of hormone? Involves a role of hormone. Which one of these? They all are having role of hormones. Yes, what is the correct option here? Okay, so yes, perfect. So over here the correct option is going to be A. Look, barrier method, no hormone. Barrier method, no hormone. Lactational amenorrhea, which is the hormone? Prolactin. High levels of prolactin. High levels of prolactin. In pills, it is progesterone or progesterone and estrogen combination. Okay, and even emergency contraceptives can be hormone IUDs, can be even pills. So that is also hormonal. Correct? Barrier method, no hormones. Copper T, no hormones. Okay, and even here barrier method, no hormones. Clear? Next, bilaterally symmetrical and acylomate animals are exemplified by bilaterally symmetrical, acylomate. This will be your biggest clue. Acylomate are all the organisms platyhelminthes before. Correct? But what are they asking you? Bilaterally symmetrical and acylomate animals are exemplified by. Which is the correct one here? Correct option here should be B. B. Platyhelminthes. B. Platyhelminthes. Okay? Because Ctenophora is radially symmetrical. Ctenophora is radially symmetrical and acylomate. Aschelminthes is pseudo-coelomate. Correct? And Annelida is going to be coelomate. Either you call it as coelomate or you may call it as eucoelomate. Okay? Ctenophora and platyhelminthes, both of them are going to be acylomate. Clear? Next, a specific recognition sequence identified by endonucleases to make cuts at specific positions within the DNA is what are those called as? Which sequence are they looking for? Endonuclease goes and looks for what? Endonuclease goes and looks for palindromes. Endonucleases are going to go and look for palindromes. What is palindrome? Malayalam. Correct? Malayalam. This way, that way, left to right, right to left is the same. You call this as palindromic nucleotide sequences. Clear, everybody? This was a simple one. Next, adenosine deaminase deficiency results into kids, whatever doubts you are having, okay, just note that in a paper next to you. Quickly note it down. Put it in the comment section. Once we are done with the session, I will personally look into it and I will try to answer all of your doubts. Okay, whatever doubts you are having, quickly write it on a paper next to you. As soon as the session ends, you will be able to comment. You will put those in the comments and I will answer you. Okay, whatever doubts you are having. So, adenosine deaminase deficiency results into what? Dysfunction of the immune system. It results into dysfunction of the immune system. The lymphocytes are not developing properly. WBCs are not developing properly. Okay. Next, which of the following RNAs is not required for synthesis of protein? Not required for protein synthesis. Tell me, everybody. Not required for protein synthesis. And yes, the correct option here is going to be siRNA. siRNA is what we are using for silencing. Okay, RNA silencing. Where are we using this? Where are we using this? Do you remember that? Do you remember applications of biotechnology? Meliodigin incognita. Yes or no? Nematode. Okay, we are silencing it. Next, when gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as, when gene targeting involving gene amplification is attempted in an individual's tissue to treat the disease, what do you call that as? You're targeting the gene involving gene amplifying. This is going to be called as none other than D, that is gene therapy. D, gene therapy. Okay, everybody? Very simple. Very simple. Very good. Next we come to DNA precipitation out of a mixture of biomolecules can be achieved by treatment with precipitating DNA. How are you going to precipitate DNA? How are you going to precipitate DNA? Yes, all the boys are going to shout out the answer to me. The DNA is going to precipitate by chilled ethanol. Chilled ethanol. Yes, everyone. DNA precipitation is happening by chilled ethanol. Perfect. DNA precipitation by chilled ethanol. Next question. Select the correct statement. Select the correct statement over here, kids. 1. Spliceosomes take part in translation. 2. Punnett square was developed by a British scientist. 3. Franklin Stahl coined the term linkage. Or 4. Transduction was discovered by S. Altman. What is the correct statement over here? Okay. So yes, the correct answer here is going to be B. Punnett square was discovered by a British scientist and spliceosomes take part in transcription. Okay, these are the enzymes which are, this is the enzyme which is going to help in splicing. What is splicing? What are you removing in splicing? Splicing Splicing means you are going to remove introns and you're going to join exons. You're going to remove the introns and you're going to join the exons. This is what we call as splicing. Okay, that is required in the transcription taking place in a eukaryotic cell, not in a prokaryotic cell. Fine. Then next, Franklin Stahl coined the term linkage. No, linkage was coined by Morgan. Linkage was coined by Morgan. And transduction was discovered by, who discovered transduction? Transduction was discovered by Norton and Joshua Eiderberg. But transduction, can you tell me who performed an experiment on transduction? Transduction experiment was performed by who? Hershey and Chase, by using bacteriophage. Yes or no? And they are the ones who gave the unequivocal proof that DNA is the genetic material. Perfect, everybody? Next, which cells of Krebs, listen everybody, very important. Which cells of Krebs of Libercun secrete antibacterial lysosome? Now you all will think, ma'am, we know that lysosome is antibacterial. But which cell of Krebs of Libercun is secreting it, kids? Which is the name of the cell? What is the name of the cell which secretes lysosome? Okay, so let us see over here. The correct answer is yes, it is B, Paneth cell. Paneth cell is going to secrete lysosome. But what about the others? What about the others? Argentopin cells are the ones which are secreting hormone. Which hormone? Serotonin. Serotonin. Zymogen cells, these are the ones which are going to be secreting the inactive enzymes. Inactive enzymes. And inactive enzymes are going to be which? Which one? Enzymes like, you can say any inactive enzymes. Zymogen basically means an inactive enzyme. Okay, so the cells which are secreting, like in the gastric cells, in the gastric glands, pepsinogen. Okay, so zymogen cells these are. Carpe cells are the ones which are present in the liver. Okay, Carpe cells are the ones which are present in the liver, macrophages. Perfect, kids. All right. So yes, the correct answer here is going to be Paneth cells. Okay. So here we go. Zymogen, chief cell, pepsin, or you can say rather pepsinogen. Pepsinogen. Argentopin cells secrete serotonin, which causes peristaltic movements. Carpe cells, liver cells, immune cells. These are the macrophages, as I told you. These are the macrophages. Okay. Next, which of the following is amino acid derived hormone? Amino acid derived hormone. Which of the following is amino acid derived hormone? Yes. So over here, the amino acid derived hormone is going to be epinephrine. Amino acid. So this is going to be, kids, if it is amino acid, means that the hormone is going to be very small. So you guys tell me, where is its receptor going to be? Receptor will be found inside. Receptor will be inside nucleus. Nucleus. Yes, receptor is going to be found inside the nucleus. Okay, so hormone receptor complex will be found inside the nucleus. Remember, epinephrine is just another name for adrenaline. Epinephrine is just another name for adrenaline. Okay, it is going to have always a stimulatory effect. Okay, also this is the panic or the shock or the stress hormone. panic or the stress hormone. Okay, next, the contraceptive Saheli. Contraceptive Saheli. What is the correct option? Please read the options very carefully. What does Saheli do? How does it work? Which one is correct? Yes? Saheli is not an IUD. Okay, it is not an IUD. It is not going to do this. It is not going to prevent ovulation. It will block estrogen receptors, preventing eggs from getting implanted. It is not a postcoital contraceptive. So correct option here, it is Saheli. And what is Saheli made out of? Centromere. Centromere. It is a once weekly, okay, once weekly pill. Clear? Non-steroidal. And it blocks estrogen receptors in the uterus. Alright, perfect. Next, what is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? What is the criterion for DNA fragments? Yes, everyone, criterion for DNA fragments, it is the smaller the fragment size, the farther it's going to move. Chotu sa hoga, it will run very fast. Correct? So the smaller the fragment size, the farther it is going to move across your gel electrophoresis. Perfect? Next, association of histone H1 with a nucleosome is indicating. H1 with nucleosome, when H1 is associating with the nucleosome, what does it indicate? When H1 is going to associate with the nucleosome, it is indicating that DNA is now going to be coiled. It is condensing into a chromatin fiber. It is going to be, yes. So kids, I would like to just let you know, stay with us. You have stayed with us in physics, botany, zoology, and guys, the best is yet to come. Next coming up is your chemistry short questions. Chemistry short questions, so don't go anywhere. Just stay here with us, short questions, so you can know that you have studied, you have prepared in the perfect way. Wasim sir is coming for your chemistry short questions right after this session. So don't even move a budge, okay? Don't budge even a bit. Chalo. So yes, the association of histone H1 with a nucleosome is indicating that the DNA is condensed into a chromatin fiber. Okay? Next, identify the vertebrate group of animals characterized by crop and gizzard in its digestive system. Crop and gizzard both are present. Which type of vertebrate animals are we talking about? Yes, perfect. The correct answer here is going to be A, and that is aves. That is aves. Aves, A is the correct option, which has crop and gizzard in its digestive system. Next, due to increasing airborne allergens and pollutants, many people in urban areas are suffering from respiratory disorders causing wheezing due to the increasing airborne. What is happening here? What is happening here? Increasing airborne. Many people are suffering from respiratory disorders causing wheezing. Why does wheezing happen? Because of the proliferation of fibrous tissues and damage of the alveolar walls. Okay? Proliferation of fibrous tissues and damage of alveolar walls. Clear, everybody? Damage of alveolar walls. Okay? So fibrous tissue is going to make, basically when fibrous tissue is going to increase, the lungs are going to become fibrous, means they're going to become harder. So breathing becomes even more difficult. Okay, they will be suffering ultimately from difficulty in breathing, tightness of chest. Okay, next. Alright everybody, let's see this. The genotypes of a husband and wife are IA, IB and IA small I recessive. Among the blood types of their children, how many different genotypes and phenotypes are possible? Tell me the answer. How many different genotypes and phenotypes are possible? Yes? So, children, if you want me to solve it quickly, we see here husband, we have husband is IA, IB. Okay, wife is IA and small I. So you have IA, children will be IA, okay, or IAI, or children will be IBI, or children will be, what did I leave? IA. So over here, the correct answer, this is blood group A, blood group A, blood group B. Okay, एक ये रहेगा I, B, and blood group AB. So, what are the correct options? 1, 2, 3, 4 types of genotypes you will have. 1, 2, 3, 4 genotypes and 3 phenotypes. So the option here, correct one will be A, 4 genotypes, 3 phenotypes. Am I clear? Am I clear? 4 genotypes and 3 phenotypes will be seen. So correct option here is A. Chalo, next question everybody. Nice, beautiful question. Chalo everybody, this is going to be a question which will tell you why exactly I have been telling you time and again that in these 3 hours, the only thing you need to maintain is your calmness. Okay, that will only be required. Look at this question. What would be the heart rate of a person if the cardiac output is 5 liters, the blood volume in the ventricles at the end of diastole is 100 ml, and at the end of ventricular systole is 50 ml. So let us see now. Let us see what all information have they given to you. First of all, let's just write the basic formula. Let's just write the basic formula where we know that cardiac output is equal to heart rate into stroke volume. Right? Cardiac output is equal to heart rate into stroke volume. What is the information they have given you here? They have given you cardiac output is 5 liters. Let me convert that 5 liters into ml. So you got 5,000. 5,000 is equal to, they are asking you the heart rate. They are asking you heart rate into. Now, what is the information they have provided you? Look, blood volume at end of diastole is 100 ml. But end of ventricular systole is 50 ml. What is end of ventricular systole means that? It means that out of 100 ml which is in the ventricle, it has only 50 ml is left behind after the ventricles have pumped. So how much has gone out? How much of blood has exited from the heart when the ventricles pumped? How much has exited? 50 ml has exited. Okay, 100 minus 50 ml is 50 ml has exited. Okay, so 50 ml is your stroke volume. So your heart rate is going to be 5,000 upon 50. That will be 100 beats per minute. 100 beats per minute. The one you needed to know here is this. And also you needed to understand. Okay, they've given you 2 values. There's a reason they've given you 2 values. Ek reason hai, listen. Volume at end of diastole means when the ventricles were filled, there was 100 ml. But after contraction, how much is left behind? How much is left behind? 50 ml is left behind. So how much came out? That is stroke volume. And that is 100 minus 50, that is 50 ml. 50 ml is coming out. So that is your stroke volume. Clear everybody? So the correct answer here is 100 beats per minute. 100 beats per minute. Chalo next. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? What is the role of RNA polymerase 3? Let's see over here. When we see the 3 polymerase, RNA polymerase. Okay, you've got 1, you've got 2, and you've got 3. This is something which is very important. They have a tendency to ask this time and again. So everyone please revise with me. RNA polymerase 1 only rRNA. Which ones? We have 28S, 18S, 5.8S. All of these 3 are RNA. Polymerase 2, HNRNA, and also mRNA. Polymerase 3, tRNA, also snRNA, and also 5S rRNA. Correct? So now what have they asked you? The role of RNA polymerase 3 in the process of transcription. Polymerase 3. What is the correct answer going to be? tRNA, also, here we go. tRNA, 5S RNA, and also snRNA. Okay? Chalo. Next everybody. Which of the following factors is responsible for formation of concentrated urine? What is helping in formation of concentrated urine? Are you guys able to see the whole question? What is helping in formation of concentrated urine? Yes? Perfect. It is going to be what your countercurrent mechanism is all set up for. It is to maintain a salty medullary interstitium, and you will call that as maintaining hyperosmolarity, increased osmolarity in the inner medullary interstitium. How much is that going to be? 1200 milliosmoles per liter. 1200 milliosmoles per liter. Clear everybody? 1200 milliosmoles per liter. High osmolarity in the inner medullary interstitium. Okay, chalo guys. Wow, you guys are doing super awesome. Super awesome you all are doing. Stay with us. I'm doing zoology. Right after me, you've got chemistry short questions coming too. Chalo, let's continue. Which of the following RNAs should be most abundant in an animal cell? Which RNA is going to be most abundant? Which RNA is going to be most abundant in an animal cell? Which RNA is going to be most abundant in an animal cell? Yes, everybody, quickly. It is going to be rRNA. rRNA will be maximum abundance. Maximum abundance. Okay? 70 to 80%. Next, which of the following is not a step in MOET? Not a step in MOET. Which of the following is not a step in MOET? Quickly tell me the answers. Not a step in MOET. Is the cow administered LH? No. Cow is going to be administered FSH like activity so that there will be super ovulation. Super ovulation. Okay. So it is not LH. It should be FSH. Okay. Clear everybody? Now a drug called heroin. Drug called heroin is synthesized by A drug called heroin is synthesized by. Drug called heroin is synthesized by what? Write out of your NCERT textbook. It is going to be acetylation of morphine. Heroin is synthesized by acetylation of morphine. Acetylation of morphine. Excellent, kids. Chalo. Next. Organ transplant rejection is prevented by which? Organ transplant rejection is prevented by. How is the speed, everyone? Everything fine? Speed is fine? Yes, the correct answer is going to be, it is prevented by immunosuppressant drugs. So as soon as the patient is given a new organ transplant is done, he will be on immunosuppressant drugs for almost nearly the rest of his life, just to make sure that his own body does not reject that organ. Okay? So yes, the correct option is D. Next question. To which barriers under innate immunity do saliva in mouth and tears from eyes belong? Which barrier? Saliva and tears. Which barrier can we classify this under? Saliva and tears are belonging to which barriers of innate immunity? Yes? Saliva and tears are going to be part of physical barrier. Sorry, just a minute. Yes, they are going to be the ones which are containing lysozymes, correct? They are going to contain lysozymes. These are antibacterial, and these are belonging to physiological barriers. Physical barriers are anatomical. Anatomical barrier means they are going to be skin and mucous membrane. Okay. Cytokine barrier are going to be your interferons. And interferons are antiviral. Okay. Cellular barriers are going to be your WBCs. Okay. Your neutrophils. Okay. Your mast cells and your macrophage. These are your cellular barriers. Clear, everyone? So correct option here is physiological barriers. Next, functional ones. Which one of the following terms would correctly describe the relationship between flight organs of animals, locust, bats, swallow and fish? Flight organs of locust, bat, flying fish. What is going to be what is it? What is the, how are you going to describe their relationship? These are all going to be performing the similar function, but their origins are different. So since their origin, here we go. Okay, I've already written it here. Sorry, kids. Since their origins are different, if anyone is still having a confusion in remembering this, HDAC. Okay, HDAC. Now, homologous. You always start with origin. So, homo means same origin. Always start with origin. Same origin and different function. Okay. Analogous. Analogous. Ana means different. So, different origin, same function. So when you see all of these, the origin is going to be different, but they're all using it for flying. So function is the same. So these are analogous organs. Clear? Homologous is showing divergent evolution and analogous is showing convergent evolution. Convergent evolution. Chalo everybody, next. What is the difference between genetic drift and change due to natural selection? Difference between genetic drift and change due to natural selection. What is the difference between genetic drift and change due to natural selection? Yes, kids? Hi, Nidha. Yes. So over here, the correct option here, kids, is going to be that genetic drift does not involve competition between members of a species. Genetic drift means sudden change, suddenly that gene from the gene pool is removed. Okay, so it does not involve itself in the further progeny. So the correct option here is B. Next, two offsprings developed in the same uterus from fertilization of two different ova. Two different ova and two offsprings. What type of twins are these giving rise to? Listen, kids, how will you solve this? How will you solve this? Two ova, okay, two sperms. Okay, two ova means two sperms. Two sperms means there can be two different types of genders. So they are two completely different ones. So they are going to be dizygotic twins, or even they can be called as fraternal twins. So the correct option here will be both B and C. Both B and C. So this means that it can be boy, it can be girl, or it can be even boy, girl. But if it is monozygotic, it is always going to be same gender because there will be only one sperm. Am I clear? Chalo, next question. Which of the following structures are not supported by incomplete cartilaginous rings? Which of the following structures are not supported by incomplete cartilaginous rings? Not supported by incomplete cartilaginous rings. Which ones? How many of you have not gone and liked our video? Just a breather in between. Yes, kids. Not supported by the incomplete rings. It will be the ones which are in the very final stage. In the very final stage just before the alveoli, and that's going to be the terminal bronchioles. The ones just before the alveoli, the terminal bronchioles, the respiratory bronchioles, the alveoli do not have cartilaginous rings. Okay, they are the ones which do not have cartilaginous rings. They are the ones which are going to have other structures which will help them prevent collapsing. For example, surfactants. Chalo, next. The main difference between DNA and RNA. What is the main difference between DNA and RNA? What is it that is different? The main difference between DNA and RNA. Main difference is going to be in the nature of sugar and pyrimidines, right? In DNA, the pyrimidine is going to be thymine, and in RNA, the pyrimidine is going to be uracil. Now, kids, how will you remember pyrimidines? Pyrimidines has a Y, and even thymine and even cytosine. They both also have Y's. So adenine and guanine become your purines, okay? Pyrimidines. Next, which of the following, and also how is the sugar different? Everyone, how is the sugar different between DNA and RNA? What is the difference in the sugar between DNA and RNA? Quickly, who will tell me? 5-carbon sugar. Yes, 5-carbon sugar. In DNA, sorry, let's write first RNA. RNA, DNA. What is the difference? Second carbon. How do you label them? First, second, third, fourth. And where is the fifth carbon? Here. Fifth carbon is over there. Fifth carbon. Correct? Where is the difference seen? This one over here will be OH. And over here it will be only H. That's why it is 2-deoxyribose. 2-deoxyribose. Is this clear? It is 2-deoxyribose. Alright. So there is a difference in the sugar and a difference in the pyrimidines. Chalo next. Which of the following would show pentaradial symmetry and have calcareous endoskeleton made up of ossicles? Very simple. The minute you come to pentaradial symmetry, it's going to be echinodermata. Radial symmetry is seen in Cnidaria, Ctenophora and Echinodermata. Okay. Mollusca bilateral in larva. Okay. And asymmetrical in adults. Okay. Annelida bilateral. Arthropoda bilateral. And Echinodermata radial. Which type of radial? Penta. 5 arms are symmetrical. Pentaradial symmetry. Clear everybody? So yes, the option is D, Echinodermata. Next, what happens when there is muscular fatigue? What happens when there is muscular fatigue? Muscular fatigue means what? What is going to be happening? There will be, in muscular fatigue, the ATP amount is low, the glycogen available is low, lactic acid is a lot. Okay, so little ATP, little glycogen and more lactic acid. Little ATP, little glycogen and more lactic acid. Yes, everybody? Alright, next. Darwin's finches of Galapagos Islands provide evidence in favor of what? Darwin's finches is providing evidence in favor of what? This is going to be, you will expect a common thing over there. You will look for adaptive radiation. But they have not given that only. Hai na? You all will look for adaptive radiation. Do you find adaptive radiation here? No, you don't find. So what are you going to look for now? Next. Biogeographical evolution. Okay, biogeographical evolution. It is nothing but you can say it is your divergent evolution on a larger scale. Divergent evolution on a larger scale. That is adaptive radiation, biogeographical evolution. Next question. The mucosal lining of which organ differs from others in concern with the number of layer of the cell? Mucosal lining of which organ is going to differ from others with number of layers of cells? Either which one is simple, which one is stratified? Nice question. Yes, mucosal lining of which organ is going to differ from others in number of layer of cell? It is going to be in the esophagus. Okay, esophagus is going to be differing. Yes, the other ones are going to be single layered. Okay, inner lining will be single layered. Okay, stomach and small intestine are going to be having what? Columnar. Okay, simple. Alright, PCT, cuboidal, brush border. simple. Esophagus, stratified. Esophagus, stratified. Okay, class? Oh, my God, very, very, very well done, kids. Kids, I'd like you to all to know, if at all anytime throughout the day you want to solve a mock test, there is always going to be a link in the description box which is mentioned below. Okay, guys, please stay tuned. Chemistry, most important questions, super short, most expected questions is coming up right after me. So stay with us. Okay, and all of you guys have worked really, really hard. I wish you all the very best tomorrow, and I will be here tomorrow. When you come out of your exam hall, we all are going to be here to see the solutions of your papers. So everyone, the minute you leave your exam hall, connect with us. We will have the solutions. You can see what are the answers immediately when you exit the hall. So everyone, immediately exiting the hall, you will be with us. We will be here live waiting for you all, and we will solve that paper together, all the subjects, PCB, everything together. Okay, everyone? Everyone, please stay calm in your exams. Okay, I wish you all the very best. Please make sure that you have worked hard. You know that you have worked hard, and you will get the result. And also always remember, at the end of the day, this is not the end of the world. Okay, kids, it is an exam. Yes, it is the most important exam of your life. Give it with a lot of attention. Please do not make silly mistakes, and only you keeping calm will avoid the silly mistakes. Please do not make silly mistakes. All right, kids? Please think twice before answering. Please make sure that every answer marked on your OMR sheet, you check the question number. Please check the question number before filling in your OMR sheets. You do not want to mess up tomorrow. Not at all. Okay, kids? Promise me you will check your question numbers before making any mark on your OMR sheet throughout the 3 hours tomorrow. Promise me, everyone on this chat box, all of you are going to promise me this. There are how many? There are 550 kids here right now. 550 kids are going to promise me on my chat box today that you are going to solve, you're going to mark the OMR sheet only after confirming the question. Everyone promise? Now, why do you want a break? No break. The short questions coming up, chemistry, right after me. Alright? Bye everyone. I wish you all the very best. Take care.